Thank you, Philippe, and thank you all for being here and the invitation to come. This is water, not gin, for the record, so don't be worried. All right, so I'm going to tell you a story today about this quite extraordinary intracellular infectious agent. I'm not sure I want to call it a pathogen because, especially in France, I believe still most of you are infected with this organism and hard to therefore call it a pathogen. But it's an extraordinary piece of biology. I will, by the way, be talking about this phenomenon at one point briefly, but when it's inside a cell, you can see already there's some extraordinary things going on, including the recruitment of host mitochondria, but there's much more to this. So this story is written by other people. I'm just the narrator, and the other people are my wonderful group, present and past, especially Mike Panis, Nicole Marino, Suchi Rastogi, Carrie Buchholz, Terrence Tyson, Magda Franco, and Michael Reese, and they really contributed all of the work that I'll be talking about today, or at least all the recent work. So who are the protagonists in this story? The first one is Toxoplasma. So this organism is a part of the phylum called Apicomplexa because it has a complexa at its api, pretty simple. And it's a single-celled eukaryote, quite a remarkable invasion machine, as you'll see. It's a close cousin of Neospora. That's important because we use that as a control. We can see, just as Etienne was talking about the different subspecies of trypanosomes and how they have evolved, we can see how Toxoplasma and Neospora have different hosts and they've evolved differently. It is, by the way, also a distant cousin of the malaria parasite Plasmodium. There are three dominant clonal types of Toxoplasma you see in Europe. In fact, I believe last I heard 90% of infections in France are one clonal type called the type 2. And you'll be seeing that, so those of you who are infected, that's what you've got, and you'll be seeing some stuff about that in a moment. So the definitive host of this infectious agent, the one in which it completes its sexual cycle, are felines. And these are various ones. This is Alain Fischer's cat. You sent me a nice picture with the mouse as a key to the life cycle. Bobcat, puma, which we have a cabin up in the hills behind Stanford, and we have one of those cameras that captures things moving by. And we see, I think we have 48 images now of these guys just prowling around looking for whatever they can find. So how does the story begin? I think it's a fun video, so let me get down here. This is from the same camera, a 10-second shot of one of these pumas, coming by, here. So walking by, and he'll begin to slow down right about here and start to hunch up his back end. And indeed, when we went, we found not this particular one, but the scat, so the feces. And in that feces may be these things, which are the oocysts that Toxoplasma forms inside the cat intestine that are shed and are super stable in the environment. So the question is, this is a classic oral fecal infectious agent. In chickens, it goes very easily from the mouth of a chicken through the gut, out the other end into the feces, and then of course for a chicken to pick that up, it's very, very easy. Felines, not so easy. They don't eat their own feces. They are not dogs. They are very fastidious. So the question is, how do you get from here back into the mouth? And the answer we know, and maybe this is actually the force that drove this, is that they're capable of infecting many of the prey that the felines feed on, and that's the fastest way back into the mouth. So this may explain why, and felines are remarkably broad predators. They're very nonspecific about what they will, as you know, they will try and eat a laser beam if you let them have a go at it. So they can eat virtually any of these things, and the parasite is incredibly broad. And yet, within certain ecosystems, there almost certainly are going to be preferences. Right? So there could be the puma and a wild turkey might be an example of one preference that you might see, or a bobcat and a possum. So as the parasite circulates predominantly in these, it may end up in others and does end up in other species, including humans. But this predominance of a particular pairing, we think, is probably driving the emergence of those three clonal types I told you about. Right? What's different about those hosts, those intermediate hosts, there's a lot that's different, and what differs the most is the innate immune responses. So if you think about immunity-related GTPases, I'll be talking about those again in a moment. These are G proteins, GTPases, that can attack membranes and destroy the membranes inside a cell within which reside intracellular pathogens. And there's various ones, both bacterial and protozoan, that they can attack. But those are found in rodents. They're not found in primates, for example. Right? So they're very specific to that. Then we have toll-like receptors. Right? So big differences in toll-like receptors, and including in humans, we lack some toll-like receptors, and we could talk maybe in discussion if there's time about why that is, and I think Toxoplasma has driven the loss of toll-like receptors in humans in some instances, nod-like receptors, etc. So there's strong selection for different effectors in these different ecosystems. So let's drill down a little bit and talk about how this whole phenomenon happens of invasion, et cetera. So here's that tachyzoid I showed you. It's the particular form we work with. And there are these organelles here that actually extend all the way up to the tip. And during invasion, a lovely image from Barbara Nichols many years ago shows that during invasion, they inject their proteins into the host cell. So those proteins are delivered across 3 membranes: the rope tree membrane, the parasite plasma membrane, the host plasma membrane, and injected into the host cell by a mechanism we still know nothing about. But they are injected very rapidly. Among the proteins they inject, and these proteins we generally call rope proteins for rope tree proteins, is a tyrosine kinase called rope16. The number's obviously unimportant, but this is a JAK2 mimic. So a kinase mimic, a host kinase mimic, that phosphorylates STAT3 and STAT6 and other stats as well, Jeroen Sai's lab has shown, in types 1 and 3, but not type 2. So the type that dominates, that 90% of French people are infected with, doesn't do this. So you see here type 1, there's an antibody to phospho-STAT3, great nuclear signal. Type 2, no. Type 3, yes. So Jeroen and Masahiro Yamamoto and the butcher in Eric Denker's lab, collectively we've all shown that this is a really remarkable phenomenon by which different strains manipulate the host response in different ways in terms of Th1, Th2 balance, and it differs between strains. Another pair, very interesting pair, is rope17 and rope18. These are now serine-threonine kinases. And they phosphorylate the IRGs that I told you are present in rodents, but not present in primates. So their type 1 has an active form of rope18, type 3 does not. So this is now yet another strain-specific difference. So the IRGs, I told you, these GTPases, there's around 20 of them in a mouse genome. And they are induced by interferon gamma. In fact, I believe they're still the most abundantly expressed mRNA after you induce with interferon gamma, and they attack the PVM. The phosphorylation of these by rope 17 and 18 inactivates them. So this is the parasite's defense against this innate immune response. And here are data from the Sibley and Howard labs that show this. So type 1 strain, the red is a marker for the parasite vacuole. Here's the host nucleus. The IRGs are sitting out here in the cytosol, and they're not able to attack the vacuolar membrane. So green is the IRG because this is being phosphorylated and inactivated. If you look in the type 3 strains, which are naturally deficient in this kinase, so somewhere in nature there's a host and prey combination that selects against this, and it's been lost in terms of activity. So the gene is not a pseudogene, but the promoter is inactive. So now you see the green of the IRGs attacking the vacuolar membrane, and they will destroy the parasite inside. And you can show that by just taking a type 3, introducing a type 1 allele of that kinase, and now the IRGs no longer attack. There's no green here, and the parasite will grow. All right, so Michael Rees in the lab was very interested in how this all plays out, and particularly interested in work that started with John Boyle and Jeroen Sai of showing that there's yet another rope tree locus, part of the same gene family. So a very extended gene family. You can see this family's been expanded massively in these organisms. And this one's a pseudokinase. So it is obviously has the kinase fold, is evolved from kinases, but is catalytically inactive. It binds the IRG and thereby altering its conformation and exposing target sites for these kinases. So this is the crystal structure, the co-structure that Michael Rees solved. And first I'm going to show you the structure unbound. So here's where is going to be the rope 5, but for the moment, just here's the structure of unbound IRG, and you'll see these two threonines here, which are the targets of rope 17 and rope 18, respectively. Watch what happens when you bind the rope 5 to this, and see how they flip out. So those two threonines flip out. Flip out when the IRG is bound by rope 5. So this is the protein that is holding the IRG and putting it into the right conformation for the punch to be delivered by the kinases that come in and thereby inactivated. And the phosphorylation here prevents this in terms of GDP exchange, as Michael showed. Very interestingly, I call this rope 5, but in fact this is a family of tandemly repeated genes, and you can see that just by counting the number of sequences. It's very hard to deconvolute this since they're almost identical. But there's between 4 and 10 in these 3 different strains, and each one's a little different from the other. So what Michael did was to map the differences in rope 5 onto the structure that he solved, and was able to show that the strain-specific differences in the rope 5s cluster up here in the binding surface that's binding to the host protein that's going to be attacking. And we believe, and the data would certainly support this, that these are adaptations to the 20 different IRGs that are present in a rodent, for example. So we have a classic arms race here. Etienne talked about one. This is a similar one where these are evolving very fast, these are evolving very fast, and they're just trying to keep up with each other in order to solve the problem of how do you get the infectious agent to sustain itself. So not only do rope trees produce these effectors, and they are injected, by the way, within the first few seconds into the host cell. They're the first thing that the host cell sees are these proteins. There are also these dense granules that sit behind these rope trees, and these will deliver their contents a little later, probably a few minutes rather than in a few seconds. They are what derive, for example, the host mitochondrial association. So if you just look staining with mitotracker, here are a couple of toxoplasma inside a vacuole. You can actually see the mitochondria even in phase. But when you stain with the mitotracker, you can see that they're surrounding them, like I showed you in that electron micrograph at the start. This is a type 1 strain. Type 2 strains don't do it. These are the parasites' mitochondria that you can see lighting up with the mitotracker. The host mitochondria are not around the vacuole. So again, this dominant strain in France completely lacks this property naturally, and yet it is phenomenally successful. Type 3 strains do it beautifully. So 1 and 3 do it, 2 doesn't do it. There's these major differences between strains. This is why what we believe, in fact, what the data clearly show experimentally, in animals at least, is responsible for the difference in virulence between these strains. So if you look at this strain S22, infecting 3 mice and imaged 1, 3, 6, 8, 10, 13 days later, and these are luciferase-expressing parasites, you can see the infection comes up, and then it's controlled. This is strain S23, given 1/10 the inoculum. It comes up and in fact is lethal, and the animals have to be sacrificed the next day or they will die. So massive differences. By the way, this and this are F1 siblings. So we can do crosses in the lab between strains, and these are brother and sister from one such cross, and their virulence, if you actually do the titration, is more than 5 logs different because of those effectors, rope 5, rope 18, and the maybe also that mitochondrial association. So some host manipulations are shared by all strains. So the major ones I've talked about so far are all strain-specific, but there are others that are common. For example, the induction of C-MYC. So here's a mock infection, and then infections with type 1, 2, and 3. Fantastic C-MYC expression. These are just loading controls. What's driving that was the question Magdalena Franco took on, and she did that in a collaboration with the Schlechtner lab in Washington University in St. Louis. And they had made mice that had GFP fused to c-MYC in the native c-MYC locus. Now, c-MYC, is, you all know, is a very potent oncoprotein. This gene is exquisitely controlled, and the only way she could replicate the induction of c-MYC using the GFP marker was to have native mice primary cells with expressed in the endogenous locus. So what she did then to find the genes that are responsible for this, she mutagenized the parasite. So here's putative mutant. And now she sorts not for the ones that turn green because they induce, but the ones that don't express the c-Myc GFP. She then clones those, sorts by sorting for the M-cherry high, GFP low, and in collaboration with the Purnam lab at the UC Santa Cruz, she was able to look at the mutants. So here they are after 4 sorts, and you can see that here's the negative control, the neospore, which doesn't turn on c-Myc. Here in green are the wild type cells, and the mutant cells, after 4 rounds of sorting, aren't showing much. After 6 rounds, they begin to show something, and after 8 rounds, they've uniformly shifted to the negative phenotype as demonstrated by the negative control here in Neospora. So now she has a mutant population. Wendy Lee sequenced those. Magda checked that they were actually defective, the ones that she sorted, and they were. This one has slight signal, but the rest of them were completely deficient compared to the wild type. And the sequencing of these 3, chosen because they came from 2 different sorts, and this one looked like it might be a slightly different phenotype, yielded, the mutagenesis we do yields around 6 to 10 mutations, coding mutations. What's interesting here, and it immediately told us the answer, was the same gene, this gene here, is showing up in all 3 mutants, each with a different mutation, each containing a nonsense mutation that kills the function of that gene, thereby demonstrating almost certainly, and Magda went on to confirm, that this is necessary, the function of that gene is necessary for the induction of c-Myc. This is what the protein looks like. It's a phosphoprotein. There's a couple of transmembrane domains, a completely novel protein, but these phosphorylation sites may be important, as I'll tell you in a few moments. Is this gene really necessary? It absolutely is. Here's the uninfected cells, and you don't see MYC induction. Wild type infection, so this is the host nucleus now, and you see great MYC induction, and the knockout you don't, and the rescue you do, and quantitation shows that this is, here's the knockout. This is a very dramatic phenotype. How does this protein function? Well, it's exported into the vacuolar membrane. So here you can see a vacuole with 4 toxoplasma inside it. And the fluorescence that we're seeing here using HA-tagged protein is predominantly around the edges, but also in between in the spaces because there's a network of membranes in there. So we think it actually is always membrane associated. The question becomes then, it's necessary for MYC induction, but is it actually the effector that drives it, or is it part of the machinery that drives it? So to address that, what Magda began to do, and then also Nicole Marino and Michael Panis, was to look at the ability of these mutants to translocate other effectors that had been described. Ali Hakimi's lab in Grenoble had found Gras-16, which is a dense granule protein necessary for an effector that drives the induction of a protein phosphatase called PT2AB, which is involved in P53 induction. And very interestingly, what Magda and Nicole and Mike were able to show is that the fluorescence that you see here in the wild type, which is the induction of this protein phosphatase, is not present when you have this knockout. And when you rescue the mutation or rescue the knockout with a wild type copy, you rescue the phenotype. So it is indeed necessary for this phenomenon. Is it necessary for the translocation? And the answer is yes. So here you see wild type nucleus. Here's the vacuole. We're staining now for Gras-16 HA tagged. Does it cross to the nucleus? Wild types it does. The knockouts it does not. So this smacks of being part of a translocon, and confirming that is a completely independent dense granule protein that Ali Hakimi's lab had identified called Gras-24. Wild types, here we have the vacuole, translocation into the nucleus. The mutants, no translocation, rescue, translocation. And we've shown this for other phenomena as well. So in fact, pretty strongly, we would say, is it effector or is it part of the delivery system? And I think it's pretty clear it's part of the delivery system. All right, but is it the complete machine? Unlikely, because these transport systems are usually quite complex, comprising many components. So we went to address this, went back to the mutant libraries and said, okay, well, the first 3 all had the same mutation, or different mutations in the same gene. What if we go a little deeper and started selecting, just screening through for mutants that had wild-type expression of the MER1 protein? These are the mutants you already heard about. And we said they must have a mutation in another gene or something. In this sense, PCR sequence show that no, this gene is fully wild-type, and they must have others. And in that way, iteratively, we've gone on to identify 2 more portions or components of this translocation machinery. This one, which we've called MER2. So here's the wild-type induction or transfer of Gras16 from the parasite vacuole into the host nucleus. Here you see in the control with MER1, I already showed you these data, we don't see translocation. And in the original mutant, the point mutant, and then in the knockout, we don't see translocation. So this protein's also necessary. It's found in the vacuole as well, seemingly more not at the vacuolar membrane, but more within the spaces between. So we think this may be possibly part of a chaperone system for moving the protein across. Further, going deeper and deeper into the mutant library, which is just, by the way, for the students in the audience, this took 2 years trying to optimize the expression for the GFP-MYC and the selection. And now that we've been milking this library for, I think, 4 years, and it's going to keep giving us more and more mutants. It's just been phenomenal from that hard work from Magda. So here's the third locus, and you can see the knockout yet again. This is Gras16HA, doesn't reach the host nucleus. Here's the wild-type control. So this becomes a third c-MYC regulation locus. Because we don't know what they do. We're just saying the phenotype is they're necessary for MYC regulation. It co-localizes with MER1. So here you can see these are the same experiment, just 2 different images. You can see here's MER3HA, here's MER1 antibody staining, and you have pretty good co-localization at the major sites, although it's not completely overlapping. And likewise here, this vacuole, there's MER3, there's MER1. So they co-localize. Do they associate? The answer is yes. Immunoprecipitation with antibodies to MER3. So the IP was anti-HA for HA tag MER3, and then immunoblot probing for whether MER1 comes down. Here's the wild type control, it doesn't. Here's the MER3 expressing HA tag pull down, and we pull down that. The protein that mediates the mitochondrial association that Lena Pernas found is not pulled down as a negative control. So they are associating. These need a little help, however. So as we went back yet again to the libraries, we found that a protein I'd already mentioned, ROP17, which is a kinase, that serine-threonine kinase, that had first been described by David Sibley's lab as being necessary for the phosphorylation of IRGs, and it indeed is also necessary for the expression of the c-MYC, the induction of c-MYC. So this is a slide I already showed you where I said, oh look, here's the same gene coming up, different mutations in it. You might have noticed, but probably not, that there's also ROP17 here. It came up, and in fact, this one is, here's the frequency of the percent change. This was a mixture of 2 different strains, both of which came through the mutagenesis. This is the one with that mutation here. This mutation in ROP17 is actually necessary, or shows that ROP17 is necessary, and we've recapitulated that with knockouts. So you see here's a wild type strain, delta ROP17. So missing that serine-threonine kinase, and you have Gras16HA expressed. The wild type, you see it goes to the host nucleus, and in the mutant, here's the host nucleus of this cell, it does not. So the kinase is also necessary. So in summary, what I've shown you is that there's extraordinary modifications going on and things happening at this vacuolar membrane. We have the mitochondrial association mediated by that mitochondrial association factor, MAF1, a completely novel protein. We've shown what the binding partner are on the mitochondria. But the dense granules release their contents into the vacuolar space by conventional secretion. So this is not an injection, but they have to get across, some of them have to get across. So these ones here, the Gras16 and Gras24, get through this complex that consists of MER1 and 3, and maybe 2, or maybe it's a chaperone. ROP17 is phosphorylating, we believe, the complex. We don't yet know which phosphorylations are necessary. And that produces the translocon that allows those proteins to reach there. These proteins which are also released by dense granules, don't need this to get into the vacuolar membrane. So this is the one that mediates mitochondrial association. This is Gra15 mediates NF-kappa B activation, which is another mechanism, as Jeroen Sai's lab has shown. So in summary, perhaps the fastest way back into a feline for an oral fecal pathogen is actually to get into the prey of there. But that's going to involve many different prey and be selection for particularly good genotypes. And an example of that is this amazing diversity of this protein, which is necessary for the inactivation of one of the innate immune defenses in a, if I can say it, a cat and mouse game that's going on between these two proteins. When we select for mutants that are deficient in the upregulation of c-Myc, we find that there's a whole collection of proteins, and they are focused here in terms of the translocon that takes them across. We think we have the effector now, but not 100% sure on that from other studies. So thanks again to the writers. Thank you for your attention, and I'll leave, and the collaborators that were so key. I think I've mentioned most along the way. Barry Schleckman's lab provided the mice that were absolutely key, Wendy Lee and Nader Permond. And I'll leave this up in case helpful for questions. Thank you.